The overall goal of this HMEJ strategy is to provide a promising genetic tool for a variety of applications, including generation of genetically modified animal models and targeted gene therapies.
This HMEJ-based method can help answer key questions in the genetic engineering field, such as how to improve the efficiency of precise targeted integration of transgenes in vivo. The main advantage of this HMEJ-based strategy is the ability to correct gene mutations with high efficiency in vivo, which further revealed its therapeutic potential. Demonstrating the procedure will be Xing Wang, a graduate student from my laboratory.
To begin the protocol, collect 5,000 N2A cells previously transfected with Cas9 sgRNA EGFP expression vectors by fluorescence-activated cell sorting, or FACS, using non-transfected cells as a control. Digest the collected cells in 2 to 5 microliters of lysis buffer at 56 degrees Celsius for 30 minutes. Then heat and activate the proteinase K at 95 degrees Celsius for 10 minutes. Amplify the sample by nested PCR using the manufacturer's protocol. Mix 1 microliter of the lysis product with DNA polymerase and a pair of outer primers recognizing the sequence around the sgRNA target site, and perform the primary PCR in a total volume of 20 microliters. Next, activate DNA polymerase at 95 degrees Celsius for 5 minutes. Then perform the primary PCR with a final extension at 72 degrees Celsius for 5 minutes.